So I'm going to start by talking about exome and whole genome sequencing and talk a little bit about who is getting sequenced now and who will get sequenced or who might get sequenced in the near future. I'm going to tell you a little bit about experiences that our group has had and other groups have had with whole genome exome sequencing, particularly empirical data about what kinds of results people want. Then I'll also show you some data about how many people will have results that might be the kinds of results that we might want to return, and how many results each person might have. Then I'm going to talk a little bit about current practices in clinical care involving exome sequencing primarily. And then I'm going to sort of make an argument about secondary results from exome and whole genome sequencing, about why I think we should offer secondary results from sequencing for return to patients and also to research participants. And finally, I'll show you a little bit of a project I've been working on here with Michael Baumschad, developing a self-directed results management tool for results from exome and whole genome sequencing called My46. So who is getting sequenced right now? And as you all probably know, exome and whole genome sequencing are really relatively new technologies, but they've had really accelerated translation both into the clinical setting and into the research setting. And right now, research participants are increasingly being sequenced, and exome sequencing in particular, and to some degree whole genome sequencing, are being used in a lot of genetic studies and in populations that have DNA for genetic analysis. And this includes studies with participants with Mendelian disorders, complex disorders, people from the general population, and also includes both adults and parents and children. In addition, sequencing is being deployed, as most of you probably know, in clinical settings, primarily to try to examine diagnostic conundrums, situations, again, primarily, although not exclusively, with children, where there's a condition that looks like it might have some genetic component, but testing to date has not yielded a diagnosis. And as I'm going to talk about a little bit later, there are currently 6 companies that offer exomes for clinical service that are being used, including at our institutions. Not all of them being used at our institution, but I'll talk a little bit about some research we've done looking at what those companies do and offer and the differences across those companies. So who may get sequenced in the future? And this is a little bit speculative, but not entirely speculative. I think there is an increasing likelihood that sequencing will be applied in some standard genetic testing settings, if for no other reason than it's going to be significantly cheaper than other existing technologies that are used to look at certain kinds of genetic testing situations and gene panels that might be ordered. I think it's possible that sequencing may be used for preconception screening, again, to try to obtain the maximum amount of information that a couple might want to have before they decide to have children. I think it's a little bit further down the road, but pre-symptomatic sequencing may happen. It may be that this is, as I'll talk about later, some information that one might get at some point in one's life to try to sort of understand what one's risks are of certain conditions before symptoms or any kind of clinical indications occur. I think sequencing will likely be an adjunct to newborn screening. There will be many situations and many conditions that are currently covered by newborn screening where sequencing will actually probably do a better job of identifying potentially newborns who have specific conditions than some of the current approaches we use. Again, there'll be a sort of cost-benefit ratio that will determine when that actually happens, but I think it will be more effective and maybe more cost-effective at some point in the near future. Several colleagues of mine here at the University of Washington actually last year published an article showing that sequencing could be used for non-invasive fetal genome sequencing using maternal blood as early as 8 weeks. I think, again, actually rolling that out into something that's used widely in the population will both need some optimization of the technique and the efficiency of the technique and the cost of the technique, but it's definitely something that can be done. And there are obviously some advantages to that as well as some challenges that come up with that. And then finally, I think sequencing may be used in the future for screening and prenatal diagnosis as well, again, for sort of the same reasons, whether it's non-invasive fetal genome sequencing or other kinds of prenatal testing. So these are the kinds of scenarios where I think we will increasingly be using sequencing in the clinical context in the near future. And so my group and my collaboration with Mike Baumschad started looking at exome and whole genome sequencing actually with the study of a genetic condition called Miller syndrome. And it was the first Mendelian disorder for which exome sequencing was used to identify the gene that caused the Mendelian disorder. And Mike Baumschad and Jay Shendure conducted this work. And Miller syndrome is an extremely rare condition, and essentially all other approaches to identifying the causal gene had failed, in part because there weren't enough families and it was very difficult to identify the gene. And basically, just to summarize, they identified the gene that caused Miller syndrome by only looking in 4 individuals individuals from 2 families using exome sequencing. So it was a really demonstration of the power of exome sequencing to identify genes for at least autosomal recessive Mendelian conditions. But I was involved in several aspects of this process, including developing the informed consent form, the informed consent process, the return of results protocol, and several other things that I won't talk about in detail today. But the reason why it's relevant to our conversation today is that in the course of studying one of the 2 families that were sequenced, the one that's shown here, the 2 adult children are the ones with Miller syndrome, and they also had a lung phenotype that had not been clearly characterized and diagnosed. It was not clear if that was part of their Miller syndrome, because Miller syndrome was so rare, it wasn't clear whether that could be part of the pathology of Miller syndrome. And through the exome sequencing, one of the genes that came up through the analysis was DNAH5, mutations in DNAH5. And basically these individuals were recessive not only, or compound heterozygotes, not only for mutations that cause Miller syndrome, but also for mutations that caused pulmonary ciliary dyskinesia, which completely fit their phenotype. And so this was an example of a situation where through the use of exome sequencing, without even looking for other kinds of secondary results, we found a result that was absolutely clinically important and significant that needed to be returned to this family. In fact, PCD is the cause of most of their clinical problems and probably will be the cause of their premature death. And so this is absolutely a clinically important result that was identified incidentally through the use of exome sequencing. And so from this first experience, we knew that thinking about how to manage secondary results, how to tell individuals about the possibility of secondary results, and providing opportunities for individuals to potentially benefit from secondary results was something that was going to be really important and we wanted to study in more detail. So I actually interviewed the participants in this study after consent and after they got results back. And the participants in this study were challenged in thinking about what kinds of results from exome and whole genome sequencing they did and didn't want to know. They didn't believe that results would substantially impact their daily lives. They couldn't really think of how it would affect their day-to-day functioning, but they still really wanted to be offered them and be given choices about them. And specifically, they said they wanted control and options for choosing different kinds of results that they wanted at different times in their life. They wanted to be able to change their preferences, so they didn't want something now, but have the option to choose to get that later. To view results at their convenience and asking questions sort of as they reviewed it potentially at their home. And to share their results with their doctor if they wanted, but they wanted to be in control of whether they shared that information with their doctor. And these results are described in more detail, and there's much more information in this article we published in the American Journal of Medical Genetics last year. There have been a few other studies that have looked at what kinds of results individuals want from exome and whole genome sequencing. And one is from a study called the ClinSeq study, which is at NIH, which is run by Les Biesecker. And they had a paper that came out last year about the intentions to receive results of individuals who are undergoing whole genome sequencing. And in their population, which was about 500 people, all participants wanted to know their sequencing results. And they had different reasons, but they included disease prevention, including the intention to potentially change their lifestyle and behaviors in response to certain kinds of results. A lot of them said something that we have heard a lot in our studies, which is knowledge is power, and that having that information allows you to decide what you want to do about that information. And a lot of them described wanting to know about results not so much for themselves, but for their children, future children, grandchildren, and extended family. And interestingly, in this study, participants distinguished between different kinds of results, but they really said they really wanted to receive all results, and they really believed that all information would be useful, even if some of the information was of unknown significance. And you could make several arguments about why they thought that, but they really wanted all information, even if some of the information was of uncertain significance. There was another paper by some of our colleagues at UBC in Vancouver that's called I Want to Know What's in Pandora's Box, Comparing Stakeholder Perspectives on Incidental Findings in Clinical Whole Genome Sequencing. And they talked to several different groups, including a couple of lay groups, parents whose children were seen in a genetics clinic, and the general public. And they found that the people they talked to wanted choice about whole genome sequencing results and incidental findings, kind of like we found. They wanted access to the raw data as part of sort of needing to respect their own autonomy and data ownership. Having control of their data was very important to them, or at least having access to their data, which is something that Malia Fullerton and Wiley Burke here have found in some other studies about research participants, but they said this very explicitly. And they associated choice and access to their data with a sense of personal responsibility to check results on an ongoing basis for updates. They saw this as sort of an engagement with data over time and part of a personal responsibility to understand what their results meant to them. And I just pulled out this one quote, which was the lay groups drew on notions of patients as partners in healthcare with responsibilities and rights of information and choice underpinned by the concept of autonomy. This reflects the broader policy lexicon of engaged partner consumers. So they really saw this as sort of a partnership and patients as partners in understanding their genomic data over time. And again, this is one subset of individuals. It may or may not be generalizable to all potential populations, but this was what this group articulated in this study. We recently conducted 16 focus groups in the King County area with parents, and we conducted them in both African Americans and non-African Americans who were primarily Caucasian. And we asked them about whole genome sequencing and about what kinds of results they would or wouldn't want to get. And we found some, I think, interesting differences between the African Americans and the non-African Americans. Among the African Americans, 64% wanted at least one kind of result, and non-African Americans, there was 80%, so slightly fewer of the African Americans. The African Americans talked a lot about distrust of medicine and providers, which has been demonstrated in the literature for this population in general. And the non-African Americans talked more about trusting their providers and really wanting them to be involved in decisions about results. The African Americans were much more concerned about the psychosocial impact of results and how it would feel to get certain results. And the non-African Americans tended to be more concerned about what they should do about results and what kinds of actions they should take. The African Americans talked about wanting community and societal benefits that would be specific to the African American community and issues of racial justice and how people think about the roles of genetics potentially in different kinds of racial identity and ancestry issues, whereas the non-African Americans didn't bring this up at all and really focused on sort of generalizable knowledge coming out of results. And finally, the African Americans really wanted and expected that whole genome sequencing results, sort of generalizable results, would be translated through their community, whereas the non-African Americans more focused on the individual or immediate family members. So sort of focused on community in the African Americans versus focused on the individual and the non-African Americans. And this paper came out earlier this year in the American Journal of Medical Genetics. So I hope from this you've sort of taken away that the data that does exist to date suggests that most individuals do want access to results, secondary results as well as primary results, and they want to be able to have control over what kinds of results they receive and when and why and how, which is sort of a challenge because we don't really have a lot of systems for actually doing that, as I'll talk about in a minute. But now I want to take a step back and talk a little bit about how many results people will likely have from exome and whole genome sequencing that might actually be something we know about that might have at least some clinical importance, if not clinical actionability for individuals. And there's a couple of papers out that have looked at this, looking in populations that have had exome and whole genome sequencing done. So there was one study, again in ClinSeq, the study I mentioned earlier, that showed that 1.4% of the 500 to 600 people they studied had a high penetrance autosomal dominant tumor or cancer syndrome variant. And they actually showed that most of these are ones that would not have been identified using other kinds of screening approaches to suggest to people that they should have genetic testing done. Now 1.4% may not sound high, but to them this was actually higher than they expected in this non-cancer population. Then a study by Refsgaard et al. That came out last year looked at genes for long QT syndrome and found that 3.2% had a published pathogenic variant in genes for long QT syndrome, which is way higher than you would expect. And this suggests possibly two things. One is that some of the published variants are in fact not pathogenic, and that some of our databases about these variants are wrong, which many of you in the audience probably know. One of the biggest challenges in genetics today is actually that their databases are evolving and often inaccurate. But it also may suggest that more people may actually walk around with long QT variants than we think, and there may be a genetic heterogeneity or other kinds of issues that affect whether or not you actually have the phenotype of long QT. So it sort of challenges our assumptions about pathogenicity. Also challenges our assumptions about what kinds of results we really will find if we look in populations and sort of seemingly healthy individuals. So we did an analysis looking at 6,500 patients from the Exome sequencing project, which was a project that was done here, among other places, funded by NHLBI under the ERA stimulus program. And these were 6,500 individuals who had a range of different phenotypes. And we looked at three main categories of variants. We looked at newborn screening as sort of a set of conditions which we know a great deal about, and we know a great deal about the pathogenic variants and how those operate for most of the conditions. And we found that 45% of the individuals had a known pathogenic newborn screening variant among these 6,500, mostly carriers, right? Not quite all, but mostly carriers. 100% had a known age-related macular degeneration risk variant in the genes for age-related macular degeneration, and we had very rigorous criteria for how we selected what these variants were. And 100% had a known pharmacogenomic variant, and we chose the most stringent level of criteria in the PharmGKB database of sort of having clear evidence of what variants actually cause changes in response to different medications. So this was sort of surprising to us to some degree, but essentially what this means is even just looking at these three categories of variants and different kinds of conditions, that 100% of the individuals in the ESP had at least one of these. Now you could argue that some people might or might not want to know about an age-related macular degeneration variant, or although it is arguably actionable, or they might not want to know about a pharmacogenetic variant, but essentially these are variants where the data is more clear. These are not variants of uncertain significance. And this is a graph showing some of the other data where we actually looked at the number of results that an individual would have. And what you can see here again is that everybody had at least one. In fact, everybody had at least 5. The blue is the African American and the green is the European American. And the average number of variants was about 15 to 16 in both the European American and African Americans, just in these three categories of genes. So if you extrapolated that out to all other potential phenotypes that you might want to look in, the number of variants that any population might have of at least clinically significant important variants might be quite high. And so this is sort of helpful. Now we have these populations with exome data. We can actually look at these frequencies and distributions and try to think about how these numbers might affect our policies and our practices about how we might offer and manage result information from exome and whole genome sequencing. And this is exome sequencing, by the way. So it's really no longer a question of whether results of clinical utility will be found with genetics or with exome sequencing and whole genome sequencing. Most individuals will have at least one clinically important disease-causing or risk allele, and most will actually have several. We think that this means that we should have a shift towards how to interpret results and how to manage results and result expectations and obligations. And in genetics and in ethics, the argument has more been about, will we find anything? And I think the advent of exome sequencing and whole genome sequencing has essentially changed the question to, how are we going to manage this and how are we going to present it to individuals? The other thing that we found, as I said, is that existing databases and knowledge about pathogenic variants are very limited. And our population-based variant frequency data that we're finding from these studies like the ESP are going to challenge assumptions about disease etiology and frequency and variant pathogenicity. And we're going to have to sort of revise how we interpret these variants as we get more of this data and understand what the frequencies are in the population and how they actually affect disease causation and progression. So in looking at how people were thinking about, okay, so you find a lot of results when you do exome sequencing. We know that there are 6 companies that are actually providing exome sequencing for clinical service right now. So we decided to actually survey those companies and to ask them about how they were approaching this. And we actually asked them about informed consent, results management, and data sharing. But I'm going to show you today just some of the data from the results management questions that we asked them and looking at their documents that they provide. And we essentially asked them these questions from the perspective of a provider or an institution about, if you're deciding which one of these exome service providers to use, how do you decide? What kinds of things would you want to know? We developed a rating system and we sort of scored them on different elements. And this paper just came out in the American Journal of Medical Genetics. And we basically found there's a very wide variability in the management of secondary results. And this includes a wide variability in the range of results that are returned, in whether or not patient preferences are assessed about secondary results and over what time course. Do you have to decide at the point at which results are returned, or do you have some time to set your preferences. There's wide variability in whether or not the result information is updated over time. There's wide variability in whether or not patients can access their results, in what kinds of informational and counseling resources are available. And the reports themselves actually vary dramatically, both in terms of how they list results and explain results, the interpretation, and even the methods that they use to generate the reports. So and this is not surprising. This is a new field and a new area and a new service that's being provided. And essentially, I think the take-home conclusion is that everybody's trying to figure this out. But right now, they're very, very different in how they're approaching this. So to sort of complicate matters, in the last month or so, two policy statements came out from ACMG. One was recommendations for reporting incidental findings from clinical exome and genome sequencing, and one was about genetic testing and screening of children, which was a policy that was created jointly with the American Academy of Pediatrics. And both of these were sort of updating older policies that ACMG had, and they had sort of a over year-long process where they actually had a committee that studied this and debated this at length and included people from very different perspectives on the issue. And it's not really fair to try to summarize both of these guideline statements in one slide, but essentially the incidental findings guidelines basically have a list of about 50 or so genes that they suggest that providers and clinicians and service providers should actually have mandatory return of result from. And they base these primarily on what they defined as sort of their definition of clinical actionability. And one of the challenges is that everybody's definition of clinical actionability is somewhat different. Not providers, service providers, clinicians, and patients. Definition of actionability in clinical genetics, I think, is much different than the definition of actionability elsewhere. So they also recommended that results for actionable adult onset conditions should be reported to children. And they were very explicit about that, and one of their arguments was that it has potential benefit to the parents, which I think is a very interesting ethical argument that actually resonates with me to some degree. If you know about something that a parent is likely to have, that the parent may not find out about otherwise, if it's something that that parent can act on and potentially prevent disease or mitigate disease or prevent negative outcomes from, that actually does have benefit to the child as well as to the adult. But this is very different than what has traditionally been recommended for genetic testing in children, both in the old guidelines in genetic testing for children and the ones that just came out. They essentially argue that genetic testing in children should be driven by the best interest of the child, and they discourage but do not necessarily prescribe predictive genetic testing for minors for adult late onset conditions, with essentially the idea that if you're going to get a test for late onset condition, you can decide whether you want to get that test when you're an adult, so that your parents shouldn't decide that for you, that there's really no advantage necessarily to having that information for most late onset conditions when you're a child. But the problem is this was really treating genetic testing sort of according to the old paradigm of genetic testing, where you have some familial risk factor, or you have some symptoms, or you have some reason why you suspect that you're at risk for condition, and you decide to get a specific test for a specific condition. Exome sequencing and whole genome sequencing don't do that anymore. You may be getting a test for a specific reason or a set of symptoms, but you have access potentially to all this other data. So you automatically get basically every other test or almost every other test that might be available. And so instead of thinking about it as getting a test and deciding to get a test, it's a conscious choice to not look at information or not allow people to have information. And I think that's essentially ethically very different, and it's practically very different as well. And these are the references for those two papers which just came out. So as I've said, there's this issue about secondary results, and I think again, thinking about sort of the old pre-exome and whole genome sequencing model, we didn't think we'd come across secondary results very often in clinical care or research, and so therefore we didn't think that there was really a strong reason to offer secondary results to patients or to research participants. But I'm just going to go briefly through some sort of point-counterpoint arguments about why we shouldn't offer secondary results, and arguments that I've heard a lot in the field over the last couple of years as people sort of struggle with this about why we shouldn't offer them. So one of the reasons I hear a lot is that we don't know how to interpret results. And if we don't know how to interpret them, then we shouldn't return them to people. And this is a picture of hieroglyphics. I actually used to have a picture for this slide of the Rosetta Stone. And I think genetics and understanding the genome is sort of analogous to thinking about the Rosetta Stone and hieroglyphics. We understand a good deal about genetics and genetic information. We don't understand everything. I think there are important debates to have about whether we should return results of uncertain significance, particularly if someone doesn't have any symptoms and we don't have have any a priori hypothesis. But it's pretty hard for me to justify not returning a result, for example, for MEN, when you know for sure that there's a particular clinical action that that patient should take to prevent the onset of cancer. It's pretty hard to argue that that's something that we don't understand and that that's something we shouldn't return. So I see this more as a process, that our knowledge is evolving and our ability to sort of decode what the genome means is evolving, but that this isn't an argument for why we shouldn't return any results at all. Another argument I hear a lot is that we don't know what results mean. And again, this is sort of, telling patients don't walk, stand still at the same time, do this, do that, or we know that you're at risk for this, but we don't necessarily have anything for you to do. And I think essentially there's a sort of underlying well-intentioned paternalism in this kind of argument, which is if I can't tell you exactly what to do, or if I can't tell you this information is absolutely going to cure your disease or mitigate your disease, then I shouldn't give it to you. And I think that has a lot of assumptions inherent in it about the values that patients have. Different patients want different kinds of information for different reasons. In genetics, we know this all the time, where a lot of information in genetics actually isn't necessarily going to change the course of disease. It's primarily diagnostic and has the benefit of having families avoid further diagnostic odysseys. So again, I don't think that not understanding everything about a result means is a categorical reason why we shouldn't offer at least some secondary results from exome and whole genome sequencing. Another argument I hear a lot is that people will be overwhelmed and upset. And I actually hear this a lot from providers, that if families are coming in for a test for one reason, their child is sick and they're getting exome sequencing done because they don't know what's wrong with them, to say, oh, by the way, I have all this other information about your child and their current and future disease risk. Do you want it? Will just be overwhelming to families. And I think it is a good point that we need to think carefully about how to present this information to people. And as you'll see, that's a lot of what my group is working on right now. But again, I think we shouldn't underestimate the ability of families to absorb lots of information at once, and we shouldn't use this as a reason not to develop tools that will actually help us present a lot or complex information to families over time. Another argument that's very common in ethics is that patients can't handle the truth. And so this is a placeholder because it's on UW-TV, I can't use a picture of Jack Nicholson from A Few Good Men. So pretend that that's Jack Nicholson from A Few Good Men. Many of you may remember that he sort of yells at Tom Cruise in the courtroom when he kind of loses it, "You can't handle the truth." And I think, again, this is sort of a well-intentioned paternalism that underlies a lot of the sort of history of genetics in general, and maybe clinical care to some degree, that families just can't handle knowing this much information, that somehow everybody will fall apart. And there actually is some empirical data now that, in fact, getting back results about various conditions does not actually cause some of the psychosocial harms that everyone thought it might. I think everyone bases their thinking about this to some degree in thinking about Huntington's disease. This is the example that most clinicians and a lot of patients and people in focus groups and interviews that we talk to bring up. And I think that's a good example of one kind of result that some families find overwhelming because they have actually seen people in their family develop Huntington's disease and because of the specific features of Huntington's disease. But I don't think we can necessarily apply that to every kind of result that families might get back. So different results will require different kinds of approaches, and we should certainly think about different reactions that families might have. But again, I don't think this is a categorical reason not to return results. And it may seem odd that I'm saying that it's put forward as a sort of categorical reason, but this is in fact what has gotten discussed in recent years as reasons not to offer these results. And finally, the main reason I hear is that it's just too hard. We don't know how to educate families. We only have a few minutes with families in the clinic. Most people don't have a basic understanding of science and genetics and risk. And we really ought to only tell them what they have to know, and this is just too hard, and our medical system is just not set up to do this. And I'm sympathetic to this argument to some degree. I mean, having spent an enormous amount of time thinking about it the last couple of years, it is hard. But again, I don't think it's impossible, and I think there are some simple ways and tools and empirical tests of those tools that we can do to try to figure out ways to have families be more engaged with the process and more engaged with the information so they can potentially benefit from that information. So if you think that then offering secondary results potentially has benefit, then we should figure out a way to offer some secondary results from exome and whole genome sequencing. The complicated question is, so then how do we do that? It's really potentially an enormous amount of information, and how do we do this in a way that's sensible, ethically appropriate, accurate, and has the potential for clinical translation? And so what we've been working on is sort of a model of thinking about instead of it being a sort of genetic testing being a transaction at a single point in time between a clinician and a patient, thinking about the genome and your genome or your patient's genome as a resource and as information about you that exists and that you can actually access at different points in time in your life, depending what your questions are and depending what your priorities are and your needs are, and that even your values about what's important may change over the course of your life. For example, if you're planning to have children soon or if you're actually pregnant, you may have certain questions related to prenatal care or related to prenatal testing and whether or not you're a carrier for certain conditions. And so that would be one set of questions you might want to know about your genome. You might want to know about conditions related to newborn screening, so then you could interrogate either your child's genome or your genome to understand what kinds of things should be looked for. You may have questions related to health about your child, where if you knew that your child was at risk, for example, for certain forms of epilepsy or for certain forms of cancer, especially if your child had actually had a de novo dominant mutation occur. This would be helpful potentially to know when your child is young, and then you could act accordingly. And then, of course, you may not want interested until later in life in conditions related to adult health, but that might be information that you might want to query about later. And so this is a picture of, a guy sitting in a library, and I don't think that's a very high-tech way of thinking about this, and I'll show you that we're developing internet tools for this. But it's essentially the idea that you want different information at different times, and that you'll come back and look for it. And when you look for it, you will get the most up-to-date information that exists about those kinds of conditions at that time. And the kinds of information that may exist about adult onset conditions may be different when you look at that than if you looked at it 5, 10, 20 years earlier. And again, the timeline is many years, perhaps over your lifetime, and then you can update this data or knowledge, which as we know and as I've talked about, is absolutely something that we're going to need to do because our knowledge is constantly evolving. So we've developed this sort of self-directed results management approach to thinking about how to manage your information from your genome. And the idea is that the individual can filter and sift results based on their own values, their own perceptions of risks and benefit, knowing that these may differ among individuals and even among all of us in this room. And temporal and contextual context, like I said, what stage of your life are you at and why are you particularly worried? Did your sister just get diagnosed with breast cancer? Did your mother just get diagnosed with colon cancer? What are the reasons why you may value certain results differently at different time points in your life? And from a sort of ethics standpoint, I think a self-directed results management model actually helps sort of facilitate the sort of 4 main principles of bioethics. The first one is autonomy, which is essentially that individuals should have control over when and whether their results are received, how they're used, and with whom they're shared at different points in time. So the individual, if they're directing their own results management, can decide that. Beneficence, which is essentially achieving maximal benefit, that by having a self-directed results management approach, you can maximize the potential benefits of genomic information according to your own values about benefits and risks, rather than someone else's values about it. Non-maleficence, which is essentially about avoiding harm, a self-directed results management model would allow you to potentially minimize the potential harms of genomic information, especially getting unexpected results or that might have adverse impact. You're not going to get something that you didn't choose to get, unwanted results or incomplete information. And finally, justice. I think a self-directed results management model, as I'll talk about in a second, may actually maximize and improve access to genomic information, might allow us to provide interpretation and translation across populations, including rural populations and non-English speaking populations and different racial ethnic populations that may have different concerns and priorities about genetics. So I'm going to show you how we do that in a second. So we recently discussed this in a paper that came out in Genetics and Medicine, but from the perspective, I think, of maybe where some of you may be coming from, I think a self-directed results management model may require a shared set of responsibilities for laboratories, for clinical providers, and for patients. And for example, how you might initiate care based on a result from exome or whole genome sequencing. I think the laboratory and the providers are responsible for sort of customary care through practice guidelines, as they already are. But the provider might initiate care for a secondary result based on knowledge of the medical context of the patient, resulting in preventive care or medical management of an existing condition or known disease risk. So you might know that your patient is at risk for familial hypercholesterolemia. You might be able to go look at genes for familial hypercholesterolemia, and you might be able to then direct management as you would in a sort of more traditional model based on those results. But at the same time, the patient might seek follow-up care or advice after receiving results on their own. They might say, I have this result, I don't know, I want to know what I can do about it. Can you help me figure out what things I should do differently in my lifestyle, or what kinds of medications or other screening approaches I should take? And patients essentially have a much greater role and autonomy than they did in the past. And I think this sort of mirrors increasing patient access to other kinds of medical information and online medical tests like you can get through your online medical record at a lot of the places where many of us get our medical care now. And I think relationships with providers will, by their very nature, need to be more interactive and more long-standing in order to engage about that information over time. And I think, again, this is sort of being predictive, but I think this kind of model could fundamentally alter the way healthcare providers and institutions use these kinds of laboratory tests and other laboratory tests, and how they interact with clinical service providers about them. If you see the information as a resource that's being accessed over time, rather than something you ask for and get in an individual transactional way. So I want to transition to sort of tell you what we're doing about this now. I've talked to you about secondary results and exome and genome sequencing, how everyone will have results of clinical importance, how everyone will have multiple results of clinical importance, and most people want those results. And so we've been developing through a couple of grants, but now through an R01 through NHGRI, a tool called My46, which is a web-based tool for managing results from exome and whole genome sequencing, but it actually can also be a tool that can be used for managing individual results from targeted testing as well. And using My46, individuals can learn about what kinds of results might be available for return and the benefits and risks of return, so they can sort of understand that information as they might in a clinical genetic counseling session now if a test was being ordered in a genetics clinic. They can actually set preferences for results return and change their preferences over time. This sense of dynamic preferences is really important and really reflects what we found from our interviews and what others have found as well. They can securely review results at their convenience and as often as they want. They can generate reports to share with doctors or family members, potentially improving translation. And there's actually an online way to query a genetic counselor via email or phone or live streaming video at any time as questions arise. So genetic counseling is still an important part of this, but some of the sort of educational and informational component actually sort of gets covered by some of this web-based tool. My46 is scalable for the volume and nature of results that will come out from exome and whole genome sequencing, and new results. For example, at the time that sequencing is conducted, we may not know that a particular result is important, and then a discovery may be made that a particular variant is important, and we would need to return that to a participant. And it can be deployed easily in large populations, including geographically diverse or remote populations. And it really standardizes and centralizes result information for participants in lay language, which I think is something that increasingly needs to be done for a lot of reasons in a lot of healthcare settings. And essentially, it allows care providers to focus on the interpretation of a result, what does that result mean for the individual, and the translation about how it should change surveillance or prevention or medical management, because it sort of takes the sort of content information about what the result is separate and allows them to focus on what the individual is going to do about the result. So I'm just going to show you a couple of snapshots of My46. We are using it right now in the R01 study that we're conducting with individuals with a large number of Mendelian and complex conditions that actually have already had their exome sequenced as part of the research studies they were involved in, and we're offering back secondary results through our study through My46. But we're also doing some usability testing as well. And so this is in development, but this is essentially part of what it looks like. And essentially, individuals can set their preferences in each of these different categories. So disease risk, carrier status, medication response, there are actually 7 different categories that individuals can set preferences in. And they can just set results for the whole category, or they can look individually at the individual kinds of results that might be available and select yes, no, or I don't know for those separate individual results. This is not a great blow up of it, but essentially you can select all yes, all no. Undecided is an effective no, but we found from our studies that people really wanted a place to indicate uncertainty, even though they can change their mind about what results they want to receive over time. One of the other things that's evolved as we've been working on this project is the sort of need to communicate information about traits as well as about genetic results. And so this is actually where individuals would see their results. And there's a trait profile, which is that circle on the left, which is the information about the particular trait or condition. And then the summary and the report and the priority actually contain the summary as lay language information about the result and what it means. The report is a technical report, which would look like what would come out from a genetic testing done in a lab right now, in a clinical lab right now. And the priority is sort of, is this high priority something that something should be done about right away, or is it medium or low priority? And that's indicated by our sort of team of geneticists and genetic counselors who review the information and update the information over time. We also have trait profiles available through the Learning Center, and this is primarily meant to be a resource for genetic counselors actually throughout the country who may be looking for information in lay language about specific genetic conditions that they may actually want to share with patients. And so we're actually having genetic counselors around the country actually write trait profiles for a range of conditions, and then we're editing them and making them available online. And each of these colors corresponds to a different category of results. And this is an example of a trait profile for DA2B distal arthrogryposis, which is a limb condition that Mike Baumschad studies. This is another picture of the Learning Center, and we've done a lot of work to try to create content about genetics and about genetic testing and about inheritance, the kind of information that people might want to know in order to decide whether they want to receive certain kinds of results or not. And we also link to many of the excellent resources that are available elsewhere, so individuals can learn more about the things that they would like to pursue further. And this is an example of the What is DNA section, and we've generated some artwork as well, and we're working on some videos to try to make accessing this information as easy as possible for people who use this, and to try to actually do usability studies to test how well people do understand this and how they interact with this information and how it affects their decision process. So just to sort of summarize, I hope I've given you some food for thought, indicating that whole genome and exome sequencing have the power to identify virtually all pathogenic coding variants in individuals who have that conducted on their DNA. They will be increasingly applied in larger patient populations and eventually in healthy at-risk populations, so I think this is not going to be something that's only done on a few people in the future. Everyone is going to have results of clinical importance, and most people will want to be offered them. They might still choose not to get them, but they want to be offered them. And I think providers and institutions are going to have ethical and fiduciary responsibilities to facilitate access to clinically important results, secondary results from exome and whole genome sequencing. I think patients will expect that if you found something that you would provide them an opportunity to learn that and understand that. And I think broader application of whole genome and exome sequencing will result in reinterpretation and increased knowledge about our understandings of pathogenicity of most risk variants. And these are going to have broader implications for genetic testing and genomic medicine and how we think about genetics influences all aspects of clinical care. I think we're going to learn more from exome and whole genome sequencing on that front, and our interpretation will be evolving and ongoing over time. I think providers are going to need to conduct research and exercise caution and consideration about which of the exome sequencing service providers to use and why. I think this is really a moving target, and I think our paper that came out recently was a snapshot, but I think this is going to be changing a lot even in the next 6 months. I think the genome is going to be used as a resource, and results will need to be offered and made accessible over time and reinterpreted to patients. And patients are going to need to be offered information and ability to make informed decisions and set preferences about what kinds of results they do and don't want. I don't think there's going to be a one-size-fits-all. I don't completely agree with the recent guidelines mandating the return of 50 results as if that's something that everybody necessarily all agrees on and all wants under all circumstances. And I think, going back to my broader point, I think just because returning and managing secondary results is hard doesn't mean that it can't be done. And I think I'm very interested, and I know a lot of other people in the field are too, in developing ways that we can actually approach these challenges and solve them. So I just want to end by saying this is a very collaborative set of projects, and I work very closely with Mike Baumschad here, and also a team of interdisciplinary people who work on genetics and genetic counseling and ethics and statistical genetics. And Julie Crouch is the research assistant who's done a lot of work with me at Children's, and Karen Dent at the University of Utah. And then a lot of the design of the site was done by Cognition Studios, and a lot of the programming was done by Catalyst IT Solutions, and there was funding from the NHGRI, the Centers for Mendelian Genetics, which is, has a center here at the University of Washington, the National Children's Study, as well as my R01. And then the data I presented from the exome sequencing project is an extraordinarily large collaborative project, of which this is just a subset of the people who contributed to the collection of the data, but without whom it would not have been possible. So thanks for listening, and I'm happy to take any questions.
Keeping the resource
Mm-hmm. So I'm supposed to repeat.
Getting that to people.
Getting that to people.
To, like, you know, if someone logs in and says, okay, now what does this mean? And that, you know, it's changed from 5 years ago.
Mm-hmm.
How does that information get disseminated to?
Yeah, that's a really good question. I've been asked to repeat your question, so even though I heard you, I'm going to repeat it for the people who are listening remotely. So the question is, how do I, and correct me if I've got this wrong, how do I envision that the information will be updated and then transmitted to people in a way that's actually useful? And you didn't say this, but I imagine also not frightening to people if an interpretation changes. I think this is one of the biggest challenges because the information, the interpretation is going to change over time. The way we are doing it right now is that essentially you have an account, and if a result changes, you receive an email that says that you have new information. And then when you log in, it clearly indicates which result that new information is for, and you can see a modified report or a new result. I think part of that is then also indicating, as part of providing that information, what the person should do, which goes back to that priority scheme. You need to go talk to your doctor about this. You need to do nothing about this. This is just sort of informative. And I think, again, this is where social science is required to actually study how people respond to this information. So that's one of the key things we're doing in our R01 is really assessing response to this information, expectations about the information in advance of getting it, and response to it, and then studying it over time, studying whether people change their preferences, studying whether they share the information with providers or with their family members. You know, it's a limited time period, so we can't update information over many years. I think it's also an open question, will people actually stay engaged with it and check it on their own? If they get that email, will they actually check it or not? So I think it's an empirical question that can be tested. But personally, I think if I have an email to check something online, and again, I'm not everybody, but I'm much more likely to do that than I am to necessarily schedule an appointment with my doctor. So I think it's trying to make that information potentially more accessible. It's not to rule out the need to talk to a doctor, but to provide a more direct way to get that information to. Sure.
So how do you link, so like do you link the information back to the person based on their nucleotide change? Or is it like a keyword? Is it, do you know what I'm saying? Like how
it's organized by trait right now. So there could be many, and there will be for most conditions, many genetic changes that might lead to a particular trait. And the report would indicate what the reports are designed to look like genetic testing reports with all the information that would be in a genetic testing report. So that would include the specific nucleotide change, the amino acid change, and what's known about the functional change, loss of function, and so forth. So, but you're asking really good questions, and not just us, but some of the other people who've been funded by the same RFA and NHGRI are really trying to figure out what we should say and what information should be provided. So it's a little bit of a moving target. So, yeah.
One of the pieces of secondary information that sort of interests me, and it's come up, I think, in our experience with HLA testing, is not finding necessarily a genetic mutation that has a disease risk, but actually finding non-paternity, which is actually, given the statistics that you gave, maybe one of the more common things that you'd actually find in doing this. Is this something that you've asked patients in those studies if they wanted to know about? Is this something that you consent, when you consent people for studies that you think, or get them for clinical studies that you think we should be telling them that we could find?
So this is a really good question. Most research studies, actually historically over the last, I'm sorry, repeat the question. Thank you, Georgia. So the question is, what about the secondary finding of non-paternity, and that this is already coming up in the context of HLA testing, and what should patients be told ahead of time and asked about whether or not they want it, and then how should it be handled? Is that a good assessment of what you asked? So this is not a new problem to either clinical genetics or to research. And for as long as I've been in genetics, which is 20 years or so, most informed consent forms have said, we will not tell you non-paternity. So if we find non-paternity, we will not tell you. Now, you could argue that non-paternity may be clinically important, but it's a sense of sort of transparency that this is just not something we're going to tell you if we figure out non-paternity, which happens a lot in linkage studies where you have families that you actually find out that the father is not really the father. There have recently been cases with array testing, and I don't know if anyone here has experienced this, where array testing has actually found not so much non-paternity as increased homozygosity, suggesting incest. Which requires sort of duty to warn and reportability issues. And there was colleagues of mine did a poster at ACMG, and I think there was a panel at ACMG about this as well. So I think the fact that non-paternity or relationships between the parents that you had not anticipated coming up in genetics is sort of a common problem. The way we're thinking about it in the context of My46 is that paternity would not be one of your options in which you would set a preference. So it would not be one of the things that would be offered for return.
how do you do that? So dad has a T, mom has a G, and the kid has a C. Does the kid just never get to know that the kid has a C?
No, well, so in the context of exome sequencing right now, if you sequence a trio, the results that are given back are usually just the offspring, not the parents. Even though the lab sequences the parents also aren't paid for through their insurance usually. It's actually through billing, I believe, for the child. And the results report that is returned is for the child. So the child would get their result and wouldn't necessarily have the parents' results to look at. Now, if the parents did get their results back, either through that testing or through some other testing, you could sit and figure it out, but that's true for blood groups too. So I think it is a problem, and it will probably come up more often, but I think it still usually requires a degree of sort of wanting to know the answer most of the time. Yeah.
So as a research study, this is really cool. There's a huge number of questions that come when you say, well, when you transfer this to a clinical model in terms of liability, in terms of risks, and I'm wondering if you've even started to think about those and where you're going. Right now, reimbursement is almost entirely linked to the wet lab, and very little reimbursement related to interpretation results.
Yep.
And also, how do you link these? Then the second question is, how do you link these up with the medical record?
Yeah.
So that once something is actionable, particularly pharmacogenetics, how can a clinician get this at the right time when the patient's potentially not coherent, let's say?
Well, those are all great questions. So to repeat the question, this is great in a research setting, but in a clinical setting, there are liability implications, there are payment implications, and there are sort of notifying the provider of medically actionable results in a medical record sort of context for ongoing medical care. And you're absolutely right, and that's all that guy scaling the face cliff of what's hard about that. We have thought about it. And in the context of the exome sequencing that's being provided right now with the 6 companies that provide it, they actually are including the billing for the interpretation that comes up in that report, at least at that single point in time, or some of them do it over 6 months to a year in the cost. So they actually are sort of including the cost in there, and the cost of exome sequencing is still fairly high. So it's also interesting for the ones that aren't providing secondary results, they're essentially not providing something that you paid for, and they're not providing access to that. They differ in how much access or whether they give access to that information over time, even if you as the provider wanted to get that information. Some of them will give it to you and some of them won't. There really aren't good mechanisms right now to actually include the raw data in the medical record at all. There are people who work on EMRs who are trying to figure out how to do that, but people haven't really figured it out yet. There are 6, and there will soon be 9 studies that are funded by NHGRI under the Clinical Sequencing Exploratory, it's CSER, I can't remember what the R stands for, where they're actually applying clinical sequencing in clinical settings, and Gail Jarvik has one here looking at colon cancer, and they actually are including the results in the medical record, but they're including the results for colon cancer, and these are all patients with colon cancer in the medical record. And to my knowledge, they have not really developed a way or a plan for actually including the secondary results in the medical record itself. So I think there's a challenge for that. In terms of liability, I think there are liability implications of not providing information about a known actionable result if you have it. And so I sort of think it's a double-edged sword, and we have to figure out how to both provide information that we know is clinically important and how to do that in an effective way. I don't have all the answers, unfortunately. So
how the My46 model fits in with that.
I think what it would be, we're working on that, and I think we're open to suggestions. It's still in early stages. But I think one model might be that it's a fee for service, and that it's sort of a subscription service over time, and that you essentially have access to that information and it gets updated over time. And this goes back to the other question that the woman asked before. You get notifications when there are changes that you might want to see, but that it's access to you, not to your clinician, which goes back to your other question. You have the information. You might be able to allow your clinician to have access to it. So one thing we've talked about is you could give your clinician permission to see as much or a little as the information as you wanted. That would depend in part on which clinician. You could allow several clinicians to have access to that information. That's a challenge now that might be met as well. But you would control who had access, and it would come to you unfiltered through your physician. So your physician might say, I'd really like you to give me access to this pharmacogenomic information because we're considering prescribing you a drug related to this. That might be part of that sort of partnership that I talked about before. But those are all, unfortunately, I don't have answers to those questions, but I'm very interested in those, and check back with me. And I may have answers later. So, yes?
I wonder if your study or other studies have looked at specific harms as endpoints and what sort of harms they might be. I mean, you can imagine you get a result for increased risk to macular degeneration, you go see your doctor about it, increased risk is identified and now you can't take disability insurance. And so
Yeah.
what sort of
No.
specific harm endpoints are you looking at?
So there are a few studies that have looked at that, not as many as you would hope. There's a the question is, have we looked at specific harms, including inability to get insurance or something like that, or inability to get employment. I think the biggest studies have been conducted by Robert Green at Harvard, looking at return of results about ApoE genotype, risk for Alzheimer's disease to adult children of elderly people with Alzheimer's disease. So that's one kind of result in one population. It may have limits of generalizability. But in general, they found that there were no adverse responses, almost at all. And that in fact, people changed their lifestyle in ways that didn't even pertain to Alzheimer's disease in a positive way in response to that information. Now that said, that's very limited. I think with other kinds of results, people may react differently. And so we actually designed what we're measuring to really try and get both at psychological and psychosocial outcomes, stigma, other kinds of things, as well as to some degree insurance and employment, but again, over a limited period of time, that's going to be hard to assess. Technically, the Genetic Information Non-Discrimination Act should protect against employment or medical insurance discrimination, but that hasn't really been tested yet in court, so I think that's sort of an open question. But we're looking at all sorts of measures of anxiety, depression, what kinds of health care you seek out, and what kinds of other testing you do. So I hope we'll have a better picture of that, as will some of our colleagues who are doing similar studies, but I think that's critical information to get in order to figure out how and whether to do this, so it's a good point.